Hello and welcome to another lecture here in Biopsychology, and today we're taking on Chapter 3, Synapses. We started off talking about the neuron, the basic structure of that. Now we're going to dive deep into the actual spot at which communication takes place over the space that we call the synapse. So here we are in Chapter 3, and one of the characters that's come into my life over the years was an individual who used to work at KVCC, and his fascination in biology happened to do with monsters, and so he had a hobby of figuring out on a realistic basis how would monsters exist. And one of the monsters that he examined was Godzilla. So if Godzilla was real, how would that happen? Like, for instance, what would he have to eat and what would his stomach have to do with it in order to produce flame coming out of his throat like he did? Just thought experiments. Now, one thing that we do know for sure, Godzilla is supposed to be really, really big. And one of the things that we recognize, and we see this sometimes in the movies, you might see Godzilla's moving around, and he's moving kind of sluggishly. And this is actually pretty real. In real life, Godzilla would be really slow. He would be really slow. He would just kind of casually move along. His nervous system has to go so much further that we would see that lag in time between when Godzilla sees the helicopter and then he tries to grab it, and it's almost guaranteed he would miss it. Just to be kind of like slowing around. Because of his size, obviously, be able to still break down buildings, and that whole flame thing, well, that'd be pretty dangerous. But nonetheless, when we're looking at the nervous system across different species, there's some variability in terms of the speed, but pretty much the limit of the size of animals in the world is partially because of the limits in the speed of the nervous system in order to navigate in the world. So you look around the world, relative to the size of the world, the animals and whatnot are all very, very small. And relative to us, let's say insects, for example, where their nervous systems have to go very, very small distances, they have way better reaction times than we do. We try to catch them or do whatever, and we can't, because their nervous system seems to be relatively quick. And actually, because it has to travel so short of a distance. So now that we've taken a little bit of time in this class to talk about Godzilla, let's talk about the science here. Now we're going to talk about the actual synapse in terms of what happens there and the physiology of it. And I want to point out here's the direction of impulse. We talked about that nerves and series of neurons all together go in one direction, other sets, in other directions and whatnot. So there's always this idea, we need to know which direction the message is moving in. And here we have this image of a terminal button on the left-hand side representing the presynaptic neuron, and then a receptor. And it's kind of shaped, in this case, like another terminal button, but it doesn't necessarily have to be. It could be right on the surface of the cell, could actually be on the axon, could be anywhere, and that's on the postsynaptic neuron. And so the information is moving, if you're looking at this, from left to right. So we have the presynaptic neuron, the electrical impulse has been traveling down there like we've already discussed. And then once it gets into this area of the terminal button, it actually allows for calcium to enter into the cell, and that creates an environment that pushes the tiny vesicles, these are little capsules containing neurotransmitters, to the surface of the presynaptic terminal button and merge with the cellular membrane and release that chemical into the synaptic gap. And the gap, let's point that out here, this gap is an actual physical space. Neurons don't touch. Just as when I was talking about the importance of glia supporting neurons, it's not like neurons are attached like a chain. We might perceive them as a chain or a rope or something, but each piece, each link in the chain would actually not be touching. They'd be just really, really close, connecting to each other chemically, not physically. So the neurotransmitters are released into the synaptic gap. In that area, those specific chemicals would then bind with particular receptors. And this is the other part here. For each kind of neurotransmitter, there is a special kind of receptor. So some neurons operate mostly with serotonin as the released neurotransmitter and serotonin receptors on the other side. Others, acetylcholine, same idea. Some others, they have all kinds of receptors, and those receptors have different functions when they get activated. But nonetheless, we have this situation where the neurotransmitters are floating across this very, very tiny gap, landing on the postsynaptic neuron and binding to the receptor. And the analogy that's been used here has always been like a lock into a key. The actual physical shape of the chemical, a property that we refer to and study in physical chemistry or stereochemistry, the actual shape of the molecule and how it, when it reaches the receptor, provides a bond that essentially unlocks a lock, unlocks a lock, and then perpetuates the opening up of ion channels that we've already discussed, and then you get that sodium-potassium thing happening, and an electrical charge begins to develop on the outside of the next neuron, and then when it reaches a particular potential, the neuron fires. So we have neurotransmitters attaching to receptors. Sometimes those receptors are activation. They will open up the ion channels. And just like I was saying before, there's different receptors on different cells. Some are inhibiting. Sometimes when there's a release of neurotransmitters and they attach to receptors on the postsynaptic neuron, it actually closes the ion channels. The neurotransmitters are then released back into the synaptic gap, and a number of things can happen. They can simply float away and dissolve into the intercellular fluid. They can be recycled and reuptake. There will be other parts of the neuron where those molecules will float, they'll bond, and then they'll reposition them. They'll take them back into the cell, put them in another vesicle to be used again. And sometimes there are enzymes in there that will actually bond and interact. And again, they sort of picture it like a pair of scissors that comes along and it cuts the actual molecule so that it doesn't fit in the key anymore, and then it's not continuing to activate. And I'll say at this point, I'm going to talk about medications a little bit later, but sometimes when we're prescribing medications in order to impact how a person's brain is working, and I guess for the rest of the body as well, sometimes we're mimicking the molecule and attaching to receptors, and we want to activate or inhibit. That might be purposeful, like we're going to put chemicals in there that will inhibit something, will keep it from happening, and then we'll put other chemicals in there to encourage things from happening. We might put in chemicals that actually interfere with that reuptake process, so there ends up being a buildup in that synaptic gap, and so it continues firing. Then we might have chemicals that we put in there that are actually those scissors cutting the neurotransmitters in half and reducing, but we might have other chemicals that are, the rock hitting the scissors. If we're going to, rock, paper, scissors, we have the rock will destroy the enzyme that is cutting, so there'll be more neurotransmitters in the gap. All of these are different ways in which we raise and lower the desired presence of a inhibiting or excitatory neurotransmitter in the synaptic gap. So there's a lot of science that goes into the kind of medicines that we prescribe, and of course, all of these are implicated in, the chemicals that we ingest, either recreation drugs or daily use kind of things like caffeine and nicotine, and then of course prescription drugs all have that sort of psychoactive effect. So these things, this is what's going on in the synapse. Okay, so let's take another look here. We have, in the synapse, we have a number of neurotransmitters. They include amino acids, monoamines, acetylcholine, neuropeptides, purines, and gases. Now, if we look at each one of these, look at some examples here to kind of bring this into some pragmatic application here. Glutamate is an excitatory amino acid. That's an example of that. Tryptophan, which tryptophan is often implicated in, it's a chemical that's, it's in a lot of foods, but we sort of think it's in turkey a lot, and because we eat turkey and we get tired after. That's actually really not the reason why we get tired. We get tired because our stomach is full, and that releases stop eating chemicals and relaxes us and whatnot. But there is a relationship between tryptophan and the production of serotonin, which can be a kind of one of the many chemicals that is released to kind of relax us and let us sleep. It's not direct. There's not, tryptophan does not cause the actual sleepiness. Acetylcholine is involved in all your motor movements. So that's a really, really big one. So if there's a drug that we're taking, so drugs, let's say, that would be for Parkinson's disease, we want to reduce the amount of acetylcholine, might be one of the treatments that we would have for that, because it would reduce the shaking movement that the individual experiences because of their Parkinson's. Neuropeptides include endorphins, and these, of course, very important in terms of pain reduction. And then we have examples of adenosine as purines and nitric oxide, which is laughing gas. These are gases that have neurotransmitter impacts in them. So what I'm going to do, I'm teaching this class, but this is really not my expertise. So I'm really depending on some of the resources in this class, and I found this really great video on gases and peptides and all kinds of different, gases and lipids, and I'm reading here the effect of carbon monoxide, different gases and stuff like that. So we're going to take a look at this movie, and it's going to go through a whole bunch of that kind of material. There's a link to this in the course book, I think, and so you can see it there as well. But I'll play it here as part of the lecture for this week.
Okay, then let's talk about the purines. The next one are all purines really are neuromodulators. So they mediate the processes of communication between two different types of neurons. And what's interesting to me is how much work probably went into figuring all this information out. But ATP actually can act as a neurotransmitter as well. That's kind of weird to me. Either excitatory or inhibitory, depending on the receptor subtype. I imagine there's a lot of things that can bind to ATP. Direct or indirect action, also depending on the receptor subtype. Found in the basal nuclei. It induces calcium wave propagation in astrocytes, hopefully by the end of my lifetime we'll know a lot more about astrocytes. And then it's also released by sensory neurons as well as injured cells to provoke a pain sensation, especially with injured cells. That's really important clinically with certain types of drugs that you give when you have like a crushing injury. I'm not going to talk about that right now, but just keep that in the back of your mind. The next type of a purine is adenosine. So if we were just to look at this whole thing right here, that's the adenosine part of it. And this is kind of a picture here showing a structure, but it's kind of a goofy looking structure. Also, neuromodulator, mostly inhibitory. It acts via a second messenger. And what's really interesting to me is that it's involved, they think that it may be involved in the sleep-wake cycle. Why? Because when you take caffeine or when you drink some tea containing theophylline or chocolate with theobromide, all these things block adenosine and therefore have systemically speaking, have this stimulatory effect. Now, I don't know why exactly I know why I mentioned it, but it's not exactly relevant to the concept of a neurotransmitter so much as it is in terms of sympathetic versus parasympathetic nervous system. But anyways, in people who have certain types of SVTs, so supraventricular tachycardia, so a tachycardia that exists above the ventricles, okay, usually at the SA node, we'll give it to people, and I don't know if they still give this to us. It's been a long time since I've worked in the healthcare, but they'll give people adenosine whenever their heart is going crazy, like this, just example, and then after adenosine, their heart will literally stop temporarily, and you might ask them to cough or something, and they'll go back to being, a normal sinus type of rhythm there. Not the best picture, I know, but anyways, that's how it works, and it works by opening up potassium channels, which immediately hyperpolarizes the cell and prevents them from contracting. All right, so the last ones that I wanted to mention are known as gases, or they are lipid solubles. Just depends on how you want to look at it. The two of them are, and this is weird to me because I always thought that these were poisons, nitric oxide and carbon monoxide. They actually are known to have an excitatory effect, usually via a second messenger, and that second messenger is usually an intracellular receptor because these guys are lipid soluble. They can diffuse through the plasma membrane, and they play a large role in the central nervous system for memory formation. So if you're remembering things, you're releasing nitric oxide. This increases the strength of certain types of synapses via a retrograde signaling. So what that means is, you have a neuron, there's the dendrites there, there's another set of, those are actually the axon termini, there's another neuron body. So whenever these guys secrete neurotransmitters and they bind here, the postsynaptic cell is going to send a signal back to the other one. And that's what we mean by retrograde signaling. So it goes from a postsynaptic cell to the presynaptic cell, sending a signal. Now, it also causes certain types of smooth muscles to contract. If you've ever talked about this right here, Viagra enhances nitric oxide. That's what we're doing here. In the peripheral nervous system, you can find it in the adrenal gland and then the nerves of the penis because of what I just talked about there. It's made on demand, and it diffuses out of the cells to the target cells because it is lipid soluble. Okay, so carbon monoxide, I haven't really mentioned that, but it has a very similar but very different pathways. So the biochemistry details of it are different, but also kind of similar. And what's in a name, really? There's more than one way to skin a cat. There's more than one way to induce these types of responses. And structurally speaking, nitric oxide and carbon monoxide are really, really similar. They're basically just like aeronucleophiles, but they're so damn toxic. I'm surprised that our brain would ever actually, synthesize these compounds. Must be some very interesting reactions we have going on inside of our cells. Okay, so the last ones are known as endocannabinoids, also lipid soluble. They tend to be inhibitory. They act via a second messenger. Don't know what happened to the words there. Involved in a lot of memory formation, appetite, neuronal development, and it actually has receptors found on the immune cells. No idea why. We know that it affects appetite. Another form of this would be THC, and otherwise known as the munchies, right? Neuronal development, all this stuff here. I don't really know how much we know about this stuff and how much of this is just correlation rather than an actual established mechanism. But anyways, it's formed by clipping the cell's own plasma membranes, and if you look at this, you can actually see that it kind of looks like the little fatty acid tails of a phospholipid bilayer. And so since it's obviously lipid soluble, it'll diffuse freely from the postsynaptic neuron. Okay, so there's that. There's another one. So this would be the presynaptic neuron. This would be the postsynaptic neuron. like other lipid-soluble neurotransmitters, and it diffuses freely from the postsynaptic neurons to the receptors, G protein coupled. On the presynaptic neurons, it's a retrograde messenger that inhibits neurotransmitter release. So once this guy receives too many neurotransmitters, it's going to send endocannabinoids from here to here saying, hey man, stop sending me neurotransmitters. So let's just talk a little bit about this. THC has actually recently been legalized, and I would like to see some better research. Because frankly speaking, if you Google anything about this shit, you go to I'mRight.com for some potheads, or you go to I'mRight.com for some people who think that weed is the devil. I don't really know much about what its effects are, because I don't know if anybody's ever been able to do really serious research with it. But now that it's being legalized, at the least, regardless of where you stand on that issue, I hope that we can get a better understanding of how this stuff works, and hopefully understand whether or not it's good or bad, and put that question to rest once and for all.
Okay, that was great. And I thought it was really interesting, the endocannabinoids and the effect of that being a retrograde messenger from the postsynaptic neuron back to the presynaptic neuron saying stop. And we might see, as there's some research that has been coming out about, I talked about Parkinson's disease, and the use of cannabis as a treatment for Parkinson's disease has shown some promise in that. And now we see the activation. We're saying, okay, stop sending me these, in this case, probably acetylcholine. Stop releasing acetylcholine from the receptor cells, probably in the muscles themselves, stop sending me these signals to react. And we can see dramatic decreases in a person's symptoms. Now, what I know mostly about in this kind of area is actually the world of psychotropics. And what this means, these are chemicals, medicines, whatever, everything that we give in order to bring about a change, or we take them or we use them in order to bring about a change in our psychiatric experience. And there's different kinds of them. Of course, there's prescription drugs that we do. This is recreational drugs. Some of them are prescription drugs that are also recreational drugs. And then everyday drugs. There's a psychotropic effect of almost every chemical that we put in our body. If you take a Pepsi, there's caffeine in it, and there's caffeine and sugar, and there's all kinds of things that create the effect that we have from having ingested that. When we take an aspirin, there's also psychotropic effects. Of course, we use these purposefully, like I was saying earlier, when we want to actually change the chemical composition of what's going on in the synapse. So that's going to be the focus of the research and whatnot that's doing, and maybe why someone will select one drug over the other. So I provided a couple examples they're really briefly outlined in the course book here. One example for each one of these. The first, looking at prescription drugs, is of course the drug fentanyl. Now fentanyl is a pain-relieving drug that is prescribed very carefully because it is incredibly powerful. It's about 20 times more powerful than codeine. And so these are very, very powerful drug, very easy to overdose on this. And the way in which fentanyl acts upon the brain is it's like our endorphins. Our endorphins are released naturally into our body when we're in pain. So just like there's neurons, purines, that are released into the bloodstream in order to activate pain neurons to tell your brain you just bumped your leg. There's also endorphins released into the bloodstream to then deaden that pain. So warning, and now we're going to deaden the pain, and you get that numbness or whatever. In fact, when we place our hand on where we hit our leg, almost instinctively we put our hand on it and we rub it, we're actually increasing the heat of the area by doing that, pulling more blood into that area and giving more endorphins into that area to relieve the pain. So that's why we rub a particular area. Now, that said, if we're going to have too much swelling, then we put ice, and that reduces the blood flow or disperses the blood so there can be less swelling and in that area. So you see, there's kind of like everyday things that we're doing that are actually activating neurons and neurochemistry in order for us to feel pain, not pain. Okay, we have too much blood in the area, let's move a little bit out. These interventions that we do that interact with the natural processes that are going on inside the body. So back to this example, both of these, the presence of fentanyl or the presence of endorphins, have the impact of providing us pleasure. This is a getting high drug, well-being, and no pain. These are, and certainly the recreational use of fentanyl is to achieve these things. Now, if you're in pain and you're not feeling well and you get a pain-relieving drug, the effect of the drug removes that pain and you feel better. Now, if you don't have pain and you take the drug, then the feel-good is the high that you get. Now, not that people who take these drugs just for pain don't also get high, but that's sort of you see that boundary between medical use and then recreational use. Now, on to our next category here, we have recreational drugs. And for this one, LSD. And it acts like serotonin. And the functions of LSD and serotonin, they change our mood, our feelings. Getting a little wonky things from my computer here, sorry about that. Eating, digestion, sleeping, and cognitive functions. Very, very much involved in serotonin. So LSD has effects on these things as well. Finally, we look at everyday drugs, and we'll pick on this one. Nicotine acts a lot like acetylcholine. Now, nicotine and acetylcholine affect our mood, our heart rate. They affect the continued release of dopamine, our reward center, and providing pleasure. So that cigarette that somebody is having is that activates the reward mechanisms. You get heart rate increases, mood changes, the pleasure and additional dopamine. So again, one of the chemical reasons why people would become addicted to that. Now, the last category here I'm going to look at is, of course, hormones. Now, hormones are released throughout the body. They affect all of the body. There are glands throughout our body that release different kinds of hormones. And it's kind of a catalog of this. We look at what the hypothalamus does, the pineal gland, the pituitary gland, the thyroid, the thymus, and then throughout the body, we have the liver, adrenal glands, kidneys, pancreas, ovaries, the placenta during pregnancy, and testes in men. And the effect of these on the body and on our cognition and our thinking and the way our brain works. So these activate neurons as well. So just kind of don't think about hormones as a neurotransmitter, but yet that's how things get communicated. That's how one part of the body is told to start growing, let's say in a thyroid situation, or changes in our mood that come from the other glands that will excrete other neurochemicals. Now that brings us down to our assessment for Chapter 3. And for assessments, we have Chapter 3 discussion, which has been canceled. It was actually about hormones, and I have decided to get rid of that one. And so the only thing for Chapter 3 is a quiz on neurotransmitters. There's 4 questions to answer about different kinds of chemicals and whatnot. Okay, that brings us to the end of Chapter 3. And go forth and do that quiz and have a great week.